Hello viewers, today we are going to learn about insulin and the topic that I am going to discuss in this video are discovery of insulin, structure of insulin, biosynthesis of insulin, regulation of insulin, degradation of insulin, metabolic effect of insulin and the mechanism of action of insulin. So first we are going to learn in short about insulin. Insulin is a hormone produced by beta cells of isles of Langerhans of pancreas. It is an anabolic hormone because it helps or promotes the synthesis of glycogen, triacylglycerol, and protein. It has found influence on the metabolism of carbohydrate, fat, and protein. This hormone is said to be connected in the development of diabetes mellitus. It is the first hormone to be isolated, purified, and synthesized. It is the first hormone to be sequenced, first hormone to be produced by recombinant DNA technology. So this much about the introduction of the insulin. So now we'll move towards the discovery of insulin. The discovery of insulin is said to be an accidental observation. It is the combination of serendipity and careful experimentation that led to the discovery of the hormone. In 1889, scientist named Oskar Minkowski, a young assistant at the Medical College of Strasbourg, and Joseph von Mering at the Hoppeseller Institute in Strasbourg wanted to know whether the pancreas of dog contain lipase or not. Lipase are the enzyme which is used in fat digestion. To resolve this issue, they began with an experiment with dog on fat digestion. They took a dog and surgically removed the pancreas from the dog. But before they could notice anything on fat digestion, they started to notice that dog was now producing far more urine than the normal dog, and the dog urine had glucose level far above normal. So these both are the common symptom of diabetes. So by this study, they came to a conclusion that lack of some pancreatic product caused diabetes. After that, Minkowski tried to prepare an extract of dog pancreas that would lower the urinary glucose level and the blood glucose level. But the insulin was degraded by some enzymes and they were unsuccessful. Until the summer of 1921, there was no significant progress on the isolation or characterization of the anti-diabetic factor. But in the December 1921, Frederick G. Banting and Charles Best from the University of Toronto were successful in preparing the purified pancreatic extract by taking proper precaution to prevent proteolysis. That pancreatic extract cured the symptom of experimental disease in dogs. On January 25, 1922, just 1 month later, their insulin preparation was injected into Leonard Thompson, a 14-year-old boy who was severely ill with diabetes mellitus. Within days, the level of ketone bodies and glucose in Thompson's urine dropped dramatically, and that extract saved his life. So, this was all about the discovery of insulin. So now we'll move to the other topic, that is the structure of insulin. Human insulin contains 51 amino acids and they are arranged in polypeptide chain. Arranged in two polypeptide chain. That two polypeptide chain are named as chain A and chain B. Chain A has 21 amino acids while the chain B has 30 amino acids. Both are held together by two interchain disulfide bridges that connect chain A at 7 to chain B at 7 and chain A at 20 to chain B at 19. So it is denoted by A7 to B7 and A20 to B19. It was the two interchain disulfide bridges. Now there is one intra-chain disulfide link in chain A between the amino acids 6 and 11. So when you take a look in the picture of insulin structure that is in the video right now, the blue is denoted with the chain A, the blue ring or the blue circles are denoted as a chain A, and the black circles are denoted as a chain B. So in this, the chain A and chain B are held together by the two disulfide linkages at A7 to B7 and A20 to B19. But when you see in the blue chain, only in the chain A, there is one another intra-chain disulfide link at A6 to A11. And apart from this chain A and chain B, there is one connecting peptide. It is also denoted. It is also denoted as C peptide. Now, the other topic is biosynthesis of insulin. Insulin is produced by the beta cell of isles of Langerhans of pancreas. The gene for the protein synthesis is located on chromosome 11, and the synthesis of this insulin involves two precursors that are pre-proinsulin and proinsulin. Pre-proinsulin is containing 108 amino acid and proinsulin contains 86 amino acid. They are sequentially degraded to form active hormone insulin and a connecting peptide C peptide. Insulin and C peptide are produced in an equimolar concentration. Whereas C peptide has no biological activity, however, its estimation in the plasma serves as a useful index for the endogenous production of insulin. Now, when you see the biosynthesis, there is pre-proinsulin at the initial, and from that, proinsulin is formed, and from that proinsulin, mature insulin is formed. So, on the left hand side, you can find out pre-proinsulin, which contains 108 amino acid. 108 amino acid. After that, you can see there is proinsulin, which contains 86 amino acid, and when there is a proinsulin formation, there is the disulfide, two interchain disulfide linkages and one intrachain disulfide linkage. And from that proinsulin, when it forms a mature insulin, that connecting peptide or the C peptide is removed. So when you see this structure of insulin or the production of insulin, the insulin and C peptide are always produced in an equimolar concentration. Because of this reason, C peptide, whereas has no biological activity, however, the estimation in the plasma serves as a useful index for the endogenous production of insulin. So now this much about the biosynthesis. Now we will move toward the next topic, that is the regulation of insulin. Human pancreas can secrete 40 to 50 units of insulin daily. So now we will discuss what are the factors that will stimulate or ignite, and what are the factors that will inhibit the secretion of insulin. First, we will discuss about the factors in stimulating or igniting the insulin secretion. This include glucose, amino acid, and gastrointestinal hormone that is present in the screen right now. These three factors, that is glucose, amino acid, and gastrointestinal hormone. Glucose is the most important stimulus for insulin release. The effects are more predominant when glucose is administered orally, or you can say that when you take a carbohydrate rich meal. A rise in the blood glucose level is a signal for insulin secretion. So in a simple word, you can just say if there is an increase in the glucose level, means there is an increase in the insulin secretion. Number two is the amino acid. Amino acid also induce the secretion of insulin. This is observed after the ingestion of protein-rich meal that causes the rise in the plasma amino acid concentration. And among the several amino acid, arginine and leucine are the potent stimulator for insulin release. Lastly, the factors that stimulate the insulin is gastrointestinal hormones like secretin, gastrin, which enhance the secretion of insulin. The GIT hormone are released after the ingestion of the food. Now we will see the factor that inhibits the insulin secretion. Mainly the thing that inhibit the insulin secretion is epinephrine, as epinephrine is the most potent inhibitor in emergency situation like stress, extreme exercise, and trauma. The nervous system stimulates adrenal medulla to release epinephrine. It suppresses insulin release and promotes energy metabolism by mobilizing energy yielding compound like glucose from liver and fatty acid from adipose tissue. So this much about the regulation of insulin secretion. Now we will move towards the degradation of the insulin. A protease enzyme, namely insulinase, that is found mainly in the liver and in the kidney, degrades the insulin. And the other topic is metabolic effect of insulin. As insulin plays an important role in the regulation of lipid, carbohydrate, and protein, insulin exerts anabolic and anti-catabolic influence in the body metabolism. So now we will see this three different topics differently. I mean to say elaborately. When you see the effects on carbohydrate metabolism, insulin influences glucose metabolism in many ways. The net effect is that insulin lowers blood glucose level by promoting its utilization and storage and by inhibiting its production. Effect on glucose uptake by tissue. Number one is effect on glucose uptake by tissue. Uptake of glucose by muscles such as skeletal, cardiac, and smooth muscle, and tissues like adipose tissues, leukocyte, and other things like leukocytes and mammary gland is dependent on insulin. So you can say that uptake of glucose is dependent on insulin by tissues like or muscles like skeletal, cardiac, smooth muscle, and adipose tissues. Whereas there are other organs like brain, kidney, erythrocytes, retina, nerve, blood vessels. It does not require insulin for the uptake of the glucose. But there is a special case in liver, with the liver, that glucose entry does not require insulin. However, insulin stimulates glucose utilization in liver and thus indirectly promotes its uptake. So it was about the glucose uptake. Now on the glucose utilization, insulin increases glycolysis in muscle and liver. The activation of certain key enzymes of glycolysis, namely glucokinase, phosphofructokinase, and pyruvate kinase, are increased by insulin. Glycogen production is enhanced by insulin by increasing the activity of the glycogen synthetase. And lastly, on the effect of glucose production, insulin decreases gluconeogenesis by suppressing enzyme pyruvate carboxylase, phosphoenol pyruvate carboxykinase, and glucose 6 phosphatase. It also inhibits glucogenolysis by inactivating the enzyme glycogen phosphorylase. So it was about the effect on carbohydrate metabolism. Now we will see the effect on lipid metabolism. The net effect of insulin on the lipid metabolism is to reduce the release of fatty acid from the stored fat and decrease the production of ketone bodies. So when you see the number one topic, the effect on lipogenesis, insulin favors the synthesis of triacyl glycerol from glucose by providing more glycerol 3-phosphate from glycolysis and NADPH from HMP shunt. Insulin increases the activity of acetyl coenzyme A carboxylase, and a key enzyme in the fatty acid synthesis. Now the next one is effect on lipolysis. Insulin decreases the activity of hormone sensitive lipase and thus reduces the release of fatty acid from stored fat in adipose tissue. The mobilization of fatty acid from liver is also decreased by insulin. In this way, insulin keeps the circulating free fatty acid under constant check. Now lastly, the effect on ketogenesis. Insulin reduces ketogenesis by decreasing the activity of HMG coenzyme A synthetase. Now the last topic on metabolism is effect on protein metabolism. Insulin is an anabolic hormone, so it stimulates the entry of amino acid into the cell and enhances the protein synthesis and reduce protein degradation. So now we'll move towards the mechanism of action of the insulin. When you see, the insulin binds to a specific plasma membrane receptor present on the target tissue, that might be muscle and adipose. This results in the series of reactions ultimately leading to a biological action. So there are three distinct mechanisms of insulin action. Number one is through signal transduction or the transduction of transmembrane signal. Number 2 is glucose transport across the membrane. And number 3 is the induction of enzyme synthesis. So now we will look about insulin receptor and mediated signal transduction. So the first topic in the signal transduction we have to know is about little bit about the insulin receptor. And once the insulin receptor is ready, you can know about the signal transduction. So when you see insulin receptor, it is on the top, that is denoted with this highlight, yellow highlight in the screen. There are four subunits of two types. Now one is alpha unit and other one is beta unit. So there are two alpha subunits, there are two beta subunits. They are held together by a disulfide linkages. The alpha subunit is totally outside the plasma membrane, or you can say as it is a extracellular, where the insulin binding takes place. The insulin binding takes place at this region, the yellow thing in the video. And the beta subunit is a transmembrane protein which is activated by the insulin. The cytoplasmic domain of the beta subunit has a tyrosine kinase activity. The insulin receptor is synthesized as a single polypeptide and cleaved to alpha and beta subunit, which are then assembled. So this was about the insulin receptor. Now when you see that signal transduction, the first mechanism of action of insulin, signal transduction, as insulin comes and binds with the receptor at this section, binds with this section, a conformation change is induced in the alpha subunit of the insulin receptor. So there is some conformational change. So this results in the generation of signal which are transduced to beta subunit. So it is transduced to the beta subunit. The insulin binding activates tyrosine kinase activity of intracellular beta subunits of insulin receptor. This causes the autophosphorylation of tyrosine residue on the beta subunit. After that, the tyrosine kinase phosphorylate the insulin receptor substrate. The phosphorylated IRS in turn promotes the activation of other protein kinases and phosphatase, finally leading to the biological action. So as you can see in the structure or in the figure, when the tyrosine kinase is phosphorylated, the IRS, you can say that insulin receptor substrate is also phosphorylated. It is also phosphorylated, and because of that, the protein kinase is activated, phosphatase is activated, leading to a biological action. So now the second one is mechanism of action by insulin mediated glucose transport. So in this, what happens is like the binding of insulin to insulin receptor signals the translocation of vesicle, the translocation of vesicle, sorry, translocation of vesicle, and this figure, don't confuse it with the changing in the slide. When the insulin comes and binds with this, uh, what happens is like the insulin receptor signals the translocation of vesicle. The insulin receptor signals the translocation of vesicle containing glucose transporter from intracellular pool to the plasma membrane over here, which is highlighted by the yellow line. The vesicle fused with the membrane recruiting the glucose transporter. The glucose transporter are responsible for the insulin mediated uptake of glucose by the cell. As the insulin level falls, the glucose transporter move away from the membrane to the intracellular pool for the storage and recycle. So it was about the second type, and now the lastly is the mechanism of action by enzyme synthesis. The insulin promotes the synthesis of enzymes such as glucokinase, phosphofructokinase, and pyruvate kinase. This is brought about by increased transcription mRNA synthesis or followed by translation, that is protein synthesis. So by this the topic is over, and in today's video we'll just go I'll just brief that we learned about the insulin, short introduction of insulin. After that we learned about the discovery of insulin, and then we learned about structure, biosynthesis, regulation, degradation, and the metabolic effect and mechanism of action of insulin. So, this might be clear with you guys. Thank you.